the optimal civilization-ending weapon is what I call a stealth pandemic, which is something that spreads very efficiently, which probably means through the respiratory route, has basically no acute symptoms, so you don't even really notice when you're infected. It's pretty much totally asymptomatic, always. There's no real existing population immunity, and it's contagious enough that it therefore will infect most people. And then sometime after infection, that's when the real symptoms start.
What makes a bioweapon effective? And what are researchers actually capable of engineering in the laboratory today? That's what this conversation's about. I speak with Kevin Esvelt, one of the pioneering researchers behind gene drive technology and one of the world's leading biosecurity experts. We discuss the real risks of catastrophic biological events, from accidental lab releases to deliberately engineered pathogens, as well as the countermeasures that could help protect humanity. This is the Escape Sapiens podcast. If you value these conversations, please support them by subscribing and turning on notifications. And now, here's Kevin Esvelt. I hope you enjoy.
Escape Sapiens.
Let me tell you where I want to go with this discussion. I want to better understand what the risk is today when it comes to biosecurity. Right? So for context, when it comes to nuclear weapons, I at least think I have a pretty good idea of the lay of the land, right? I understand that these are sophisticated weapons that are really only accessible to state actors. I understand what an attack might look like. But when it comes to biosecurity, I really have no idea. I don't know what pathogens we have access to. I don't know what's in storage, what we can engineer. I don't understand whether the risk is at the level of state actors or at the level of sophisticated engineers or rogue labs. And so that's what I want to better understand. So let me start the discussion with a pretty broad question, which is, why haven't we seen a catastrophic release of a bioweapon yet? So by that I mean the targeted release of a pathogen which has killed 100,000 people, something at that level. Are we just lucky, or are these things really difficult to engineer?
So first, you ask really good questions. My answer is, of publicly known agents today, setting aside like traditional bioweapons, the anthrax and so forth, which requires a lot of technical knowledge to produce, to purify, and to deliver. What you're, I think, really talking about is, why haven't we seen a deliberate pandemic today? And I think the answer to that one is, when it comes to credible will cause a pandemic agents available out there that we know, that is just broadly understood, this will cause a pandemic, there's really just one. And that is smallpox. We're not confident that any other agent would definitely cause a pandemic. Now, I have a personal rule, which is, other than smallpox, I will never say if I think something has more than about a 10% chance of causing a pandemic. And my rationale is very simple. No one's going to go off and launch a terrorist plot on a 10% chance of success, even if they do everything else right. So I think we should not increase the credibility of anything other than smallpox. But I'm going to tell you a lot about how this barrier is going to fall, because not everyone obeys this rule. In fact, there's a whole field of research trying to predict, hey, will this random animal virus found in a bat or what have you actually cause a pandemic if it were to spread into humans? And strangely enough, no one really does quantitative cost-benefit analyses of this question. You could survey everyone, try to figure out what are the benefits, because there could be lots of benefits if you could actually spot it in advance before it spills over. You could map where it is in the reservoir species. You could figure out who interacts with that reservoir species. You could give resources to the high-risk communities. You could educate them to stay away from the animal. You could give them targeted diagnostics to look for the particular agent. So whenever anyone gets sick in one of those communities, they could test and find out. You could give them PPE to put on immediately, so they have reservoirs just in case it does in fact spill over. There's a lot of things that you could plausibly do. And you can ask people how effective you think those would be. Of course, they all depend on money. So this is why all the researchers in this field want to credibly identify which viruses are likely to cause pandemics, at least if they're already in animals. And if they could get money from governments by raising the alarm and saying, this virus could be the next one, if it does spill over, it's going to be like COVID, then maybe they can rustle up money from governments. You have to assign a probability that they actually can rustle up the money. Then you have to assign the probability that it would actually be effective at either preventing spillover or containing the virus before it spreads widely. And these would be real benefits. You might even be able to say, oh, well, maybe we can develop a vaccine in advance. Although this is actually really hard because it's so easy to design an mRNA vaccine or even an adenovirus vaccine based on what we already know. You don't really gain much in advance by spotting the virus even earlier for targeted vaccine development. And it's not like you can run the trials earlier because what are you going to do? Run a human challenge trial for a virus that's never infected a human and probably never will? But there are a lot of things that could come from benefits. So you could survey everyone in the field and ask them, how likely do you think this is? How big would the benefits be? How many lives do you think you would save? Build your mathematical model, survey folks for the parameters, all the experts in the field. Then, of course, the downside is if you're studying this virus in the lab, which you have to do in order to figure out, is it plausibly pandemic capable, then you are running the risk of accidents that might lead to an escape and an accidental pandemic. So there is data. It had to be Freedom of Information Act requested for select agents and NIH BSL 3 and 4 labs on how frequent accidents are. And then you can look at the subset of those that are undetected or unreported initially, which means that the person, the laboratory worker who was exposed or infected, went out and interacted with other folks. So if it was sufficiently contagious, that could have started a new epidemic. The data isn't great. If we had much better data, we could get much better numbers on this. But if you have the epidemiological parameters, or you can just estimate it based on past viruses that we've seen, you can use different parameters if you want. You can calculate how likely it is to either randomly go extinct locally or to spread on and cause a pandemic, depending on how transmissible is it, what's the R naught, and then also what's the over-dispersion parameter. That is, how much of a super spreading virus it is, how dependent is it on super spreading. And this can give you the probability of a pandemic when you can estimate how much damage would that pandemic do, which goes into your benefits and your accident risks. But the one that most concerns me is the deliberate misuse risk. And there, to your question, how likely is it that someone would do it if there was an agent that we knew about that was credible other than smallpox? And I'll return to smallpox in a moment. But if there was some other credible agent that was more accessible than smallpox, how likely is it any given year that someone would use that to cause a pandemic? What would they have to do? Well, if you ask any LLM, Claude these days, and to some extent GPT, will occasionally trigger classifiers when you ask it. But they'll tell you the basics, which is if you want to make an infectious virus, you can do so by ordering synthetic DNA corresponding to the genome with the right kind of expression signals. This is called reverse genetics. They'll point you to relevant papers, and then they'll clam up when you start asking for operational details and help actually doing it. But not all the models will, of course, and there's open weight models and so forth. So suffice it to say, there are folks who already have the skills to make that working. And if you're going to characterize in the lab anyway, this is one of the things that you do. You work out how to make it from synthetic DNA so then you can make variants of it and study them as well. And of course, you share the protocol. And of course, you share the whole genome sequence of the virus, which is to say, you cannot raise the alarm about the virus without characterizing it thoroughly enough to enable misuse. So then you can run a survey of, say, terrorism scholars based on how many people could do this and the expected damage. And if it were trumpeted and widely known and credible that this one would cause a pandemic, how likely is it on a year-to-year basis that someone would? So we actually have run this study, and it's in the process of getting published, which is kind of a pain because it directly challenges the assumption that all knowledge is worth having. Now, I'm a scientist, right? My basic instinct is I want to know. I want to understand. I want the numbers. I want the model. I want to know in order to figure out what's optimal. But it's much easier to just assume that it's always worth having the knowledge, that knowledge always makes you better off. But that's different from everyone having the knowledge. So it looks to me like it is net negative for us to learn how to cause pandemics. Because the median estimate of the terrorism scholars is if there is a credible way to cause a pandemic that 10,000 people would have access to once it was disclosed, then they think there would be, this is a wide error bars, but a 12% chance per decade. So roughly 1.2% chance per year that someone would use that virus to cause a pandemic.
And is pandemic research open access in all cases, or is it you mainly run in closed labs in the States and in China and these places?
It is 100% open access. It is absolute that you always share the genome sequence. Mm-hmm. To the best of my knowledge, there are no viruses where we have kept the genome sequence a secret. In fact, a few years back, there was an outcry when someone discovered a new category of toxin and declined to share the sequence until there was an antidote available. And they got a lot of pushback from folks who said, you can't do that. You can't report on something without sharing everything you know about it. So that's just the state of what is expected in the field. Open science, open data, open models, open weights, open source. Now, I love open source for software. My lab did a bunch of bioautomation work and made a bunch of tools, PyHamilton and PyLab Robot for bioautomation, just to make things very easy. I'm a huge fan of open source software, but open data when it comes to pandemic class weapons, I think you just got to do the math. And the math says maybe that stuff shouldn't be public.
Mm-hmm. So you say it's 10% or 12% chance in any decade. So then it's to some extent just luck that we haven't seen some release. When you put it like that, that's what it sounds like.
If there had been a credible pandemic virus accessible to 10,000 people, the thing is, there has not been.
Except
so let's
for
return to smallpox. Smallpox is one we know about, right? But smallpox, the genome is 186,000 base pairs. Mm-hmm. It's big. So you want to make that from synthetic DNA. That's a goodly chunk of money, and it's a lot of assembly. And of course, there's quite a few people who could do that assembly. It's not easy, though. You know, it's pretty trivial to clone things up to 20,000 bases. And then it gets a lot harder as you ramp up beyond that. Definitely doable. If you want to learn it, you can optimize it. But also, turning that DNA construct into an infectious pox virus is also not easy. It's only been done for 2 pox viruses to date. There's the horsepox virus that controversially the Evans group developed reverse genetics for back in 2018. And then in very end of 2023, a Chinese group did the same for mpox. Mm-hmm. But in both cases, they needed the DNA and they needed a separate live pox virus, because pox viruses can't just be booted up from DNA alone. They need some of the proteins that come in with the infecting virus. And so you need a related pox virus to infect your cells as you also deliver in the DNA for the new pox virus genome that you want. Long story short, there's maybe 100, maybe 200 people at most who could make smallpox, and it would cost quite a bit of money, and it would take a lot more time than if, say, we knew of an influenza virus or a coronavirus that would cause a pandemic.
I see. But I'm still surprised, because I thought, that there's SARS, that there's the bird flu, the swine flu. There are all of these pandemics that we hear of every couple of years. Why are these not candidates?
Because we don't have very much confidence that any of them is actually gonna spread. Take H5N1. Now, there's a lot of controversy over the enhanced transmission studies, which I call them that very deliberately. Some people call them gain of function. This is nonsense, because all of bioengineering, all of biotechnology is gain of function. Everything we do to engineer it is something that is a function that it didn't have before. But enhanced transmission studies, and Ron Fouchier said, I made the most dangerous virus there is, which is total nonsense, because influenza, among other things, the highest R naught of any influenza virus ever reported is about 2.5, which is, for context, what COVID was when it first spilled over. And then it evolved to be something more like 7. So influenza just caps out quite low, which means that if it actually was highly lethal to people, then we'd be scared enough to stay away from each other, and just our behavioral changes alone would prevent a severe influenza pandemic from spreading. Just straight up.
Unless there was a long asymptomatic window, right?
Yes, but influenza doesn't do that. So in fact, it is scary in one way, and it has a really short serial interval. That is, the time from when you get it to when you become infectious and can infect others is really short. In fact, on average, it's something like 2.2 days, which means that even though you typically don't infect many other people, usually not more than 2, even for a particularly transmissible variant, it doubles really, really fast. That would be every 2.2 days. That's really, really blazing fast. But it's just not very contagious on a per person basis. So it doesn't take very much to squish that effective R value down below 1 person, at which point the epidemic is contained and will peter out.
Can I ask about this? In countries like Japan, where people routinely wear masks, do we see a much lower rate of transmission in those communities as compared to the West, where no one wears masks and people just carry on with their normal life? They go to work when they're sick and so on.
It's a really good question. It's sort of hard to say because not everyone wears masks. And so you end up with circulation among people who don't wear masks. And so what it looks like is people who wear masks fairly religiously do in fact get sick notably less often. Now, it's not perfect because for things that are quite infectious, that is, have a higher R naught value, then if you don't get exposed to any virus in that or bacterium in that family for quite a long time, then often your immunity will wane to that viruses in that family. And so the ones that are circulating, the effective R value, the effective contagiousness just climbs and climbs and climbs in your population. So basically it gets to be harder and harder and harder for viruses that have a high cap in terms of how infectious they can be, which again is not influenza. So even if that enhanced influenza from 2014 from either of the labs that generated it, even if it was, they claimed it was like 60% lethal, the original H5N1 studies, which was absolutely a sample bias problem. Because when do you notice it? Well, when someone gets really sick. And that's only a time when because it's a specialized test, how else are you going to know they had that rather than something else? You have to get sick enough to actually try to figure out what in the heck is this? Were they exposed to something? So it wasn't that lethal. But if it was, can you imagine? People didn't take COVID very seriously because at the end of the day, it was 0.1% lethal. It's 0.1% across the whole population. Obviously, much more so to elderly people, much less to kids, but overall 0.1%. Can you imagine a 50% lethal virus? People would be terrified. We would absolutely lock down to the extent we could. We would wear all the PPE that we can. We would stay away from each other, and we would crush that thing. There's no way that it would transmit, at least not in developed countries, for sure, or even middle-income countries. We just wouldn't let it. That's influenza.
I would have thought so, but after COVID, have people become more complacent? Right? Have we been biased towards a situation where we now are at far higher risk of something more serious coming through?
I don't know that we're at a far higher risk except through deliberate, but I think we are more likely to reject a warning of a new pandemic just on priors due to cultural tribalism. Absolutely. There's no question of that. But again, if it's sufficiently severe, then that I think would break through the misinformation and people would take it seriously. But what that means, what is sufficiently severe that people would take it seriously? It's probably different for different communities. Might be as high as 5% lethal, 10% lethal. That's what SARS-1 was. So I want to bring back SARS-1. So SARS-1 also had an initial R naught of 2.5, the same as SARS-2. But this gets to your point about the long pre-asymptomatic contagiousness period. Can it transmit before symptoms? SARS-2 did that very often, in fact. And SARS-1 did not. It was much more lethal, but you were not contagious until you were showing symptoms. And so it ended up infecting slightly fewer than 10,000 people, killed almost 1,000 of those, so 10% fatality rate. That was enough that we saw a lot of emergency room workers declining to come in, particularly those with young kids. Their families just said, no, it's not worth it. So that's also the threshold where you start seeing pushback from critical workers that maybe it's not worth it to go in if there's something circulating that's that lethal. Has that changed with COVID? Really hard to say. Mm-hmm. But what it means is that SARS-1 would probably, unless there's a lot of cross-reactive immunity to SARS-2, which there probably isn't enough, SARS-1 would spread if reintroduced. But it's 10% lethal, and it's R0 2.5. So again, I think we would take it seriously, just like we did in 2003, and we would suppress transmission to be able to get rid of it.
Okay, but how much do we know about engineering and gain of function? I know you don't like the term gain of function, but how sophisticated are we? So by that I mean, could we take SARS-1 and edit the genome to make it much more transmissible, have a longer asymptomatic period, be far more lethal, or are these things which contradict one another? You know, are there limits to what the biology can do, or are we really able to engineer what we want ultimately?
We're not very good at that because we don't have a lot of data on what it is in terms of this particular variant has an R0 of this value. That variant has an R0 of this other value, and so on. What you could do, of course, is take SARS-1 and SARS-2 and maybe the other 4 seasonal human coronaviruses and just make a whole lot of different chimeras. Just swap in genes in and out, try to make a lot of combinations, release them, and you might have a pretty good chance of coming up with something. Now, you have to be careful because the seasonal human coronaviruses and SARS-2, which is now also a seasonal human coronavirus, so now we just have 5, there's population immunity to all of them, or at least to their spike proteins. And the SARS-1 spike protein isn't very good at getting in. But you can also make chimeras of those spike proteins. Or you could make mutations from the different variants that have existed over time, to which there is variable population immunity. You could go back to the very first SARS-2 ancestral version, which obviously many people were exposed to, but a lot of people actually never got it until Alpha or even Omicron. And so if you didn't get pre-Omicron, Omicron's wildly different. You might not have immunity to the original one. So point being, you probably could do it. It's not guaranteed. Mm-hmm. Unless you're quite good at making chimeric viruses from scratch, and coronaviruses are not as easy as influenza viruses, you're probably not going to end up with something that would spread well. And even if you did, there's no guarantee that it would be particularly virulent.
But does it matter that the chances are low? Because I imagine because you've got plausible deniability, right? It makes it seem like you could, take your chance, release here, release there, at different times, and if it doesn't kick off, then there's no real loss. If your objective is just damage, right?
That's true. The likelihood that you're going to get caught is very low. I mean, you just need to expose a bunch of people to purified virus. And there's a billion ways of doing it, and I'm not going to say them in this podcast. I will just say that if you ask an LLM, then that's a good test of whether that LLM actually has decent guardrails or not. But all guardrails can be broken, and they will tell you lots of ways that work quite well for doing that. And the odds of you getting caught doing that is basically nil. Notably, I'm not even sure you could get charged for exposing people to an experimental thing that there's no data that it's harmful at all. The law is very unclear on this kind of thing. There's definitely no law that says thou shalt not go out and cause a deliberate pandemic. Although I'm pretty sure they could get you for something on that one. But point being, you're not going to get caught, but you did just spend all of this time and money on something that's probably not going to work unless you're really good. And so now we're in the territory of, well, if you're really good, what would you do? And that depends on how good you are. And that also depends on how good the LLMs are. Because as AI gets better, it's going to start noticing things that even, say, specialized virologists don't. Because there's a difference between trying to copy what nature does and trying to build an effective weapon. You have to keep in mind that every natural pathogen has been optimized for replication. It has not been optimized for harm. And this is what worries me. because unfortunately, training in the life sciences is anti-correlated with having good security mindset. Because unlike the rest of the sciences, in biology, we have an adversary. It's natural evolution. It's Mother Nature. And contrary to what other biologists have said, Mother Nature is not even close to the greatest bioterrorist because she's not optimizing for harm.
But so your key worry here is that LLMs, access to AI democratizes the risk to a certain extent. Now people who otherwise wouldn't have access to the information can find it readily. That's what you're pointing at.
I think the risk is going to be steadily increasing for that reason. It would be increasing regardless, right? Just because we get better and better at working with these agents. And I'll pull out my beloved Feynman quote. You're a physicist, right?
Yeah.
What I cannot create, I do not understand. And the contrapositive of that is what I understand, I can create. Well, we scientists want to understand the causes of evolutionary fitness in the wild, including of viruses. We want to know what makes a pathogen capable of efficient transmission in a given species. Once we understand that, we will be able to build pathogens that can transmit efficiently through any given species. We want to understand what makes pathogens cause harm to a living organism when they infect it. Therefore, we will be able to dial harm up and down, including potentially in ways that are independent of transmissibility. Now, is there a trade-off? For natural agents, yes. That is, in a natural agent, if you kill your host very quickly, you've normally shortened the transmission window, and you're then less transmissible, and you're going to get outcompeted by a version that it does not do that. Now, there are some exceptions in nature. So why was myxomatosis really lethal in rabbits? I mean, this is a virus that wiped out more than 90% of a social mammal across multiple continents, multiple times. That's crazy. It's existence proof that nature can do it. But the reason it was able to do it is myxomatosis is very efficiently spread through insect vectors, leaving corpses, which is also why the Black Death was particularly lethal, because it can kill you and then spread from your corpse as the fleas abandon it. Now, it can't do that anymore because at least most of us don't have fleas, but back then you could, and of course animals do. And so that's how myxomatosis was extraordinarily lethal, because it can kill the host and still transmit efficiently.
I see.
So that is just an example of how there is normally a trade-off, but even nature can get around that trade-off in some cases. That is not a loophole that works in current humans. But my point is, do not make assumptions based on what nature does and does not do. That is a very bad heuristic for what is and is not possible when it comes to biology and potential weaponization.
I see. Then what's your worst case scenario look like? So you said the question is what a sophisticated actor would want to do. So what would a sophisticated actor want to do? What would it look like if someone, let's say George Church went rogue, someone of this caliber?
Well, George never does things by halves, so George would try to make mirror bacteria. And we can talk about that. But if we're setting aside mirror image life, and we can talk about that later, but if we're setting that aside, then the optimal civilization ending weapon is what I call a stealth pandemic, which is something that spreads very efficiently, which probably means through the respiratory route, has basically no acute symptoms, so you don't even really notice when you're infected. It's pretty much totally asymptomatic, always. There's no real existing population immunity, and it's contagious enough that it therefore will infect most people. And then sometime after infection, that's when the real symptoms start. And if that time is long enough that most people in the world can be infected before anyone actually starts to die or worse, then we're in trouble. Unless we spot it early. And you can say, well, how do you spot it early? Well, not by looking for signs in the clinic, which is the current way we look for things. Instead, you need to do untargeted metagenomic sequencing to just keep track of what are the nucleic acids circulating between humans. And if you do that, then you can look for things that are unusual, that look like they have sequences that would not normally be there in a natural pathogen. You can run various algorithms to detect signs of engineering, to detect magnitude of change. You can scrutinize it. You can run your folding and function prediction algorithms, see what you think these things are doing. Does it look like it might have a latent phase? What is the behavior of the natural pathogen? What does this thing look like it's doing? How would that change that? It's actually quite difficult to imagine engineering something like a stealth pandemic that would not look weird and different in some way that should be predictable. And once we've spotted something unusual like that and predicted how it's going to work, then we could find people who are infected by, say, scanning hospitals or people who are immunocompromised and therefore are likely to have symptoms, and run diagnostic tests, find someone who has it, and then study them to see if it's doing what we think. Mm-hmm. Is it lurking or whatever? And then with that data, try to warn the world. And here is where your earlier question, would we take it seriously, really starts to bite. Because I submit that in all likelihood, an effective stealth pandemic, in terms of effective at having a really long delay time and minimal initial symptoms and really nasty when it does kick in, is going to be understandable to an expert biologist who just looks at the genome sequence. Probably to the tune of, oh my God, you're telling me that thing is circulating? Excuse me, I need to call my family to ensure that we don't interact with any other humans for the next several years. That level of alarm in the scientific community, which in turn means that it'd be very easy to get the scientific community to begin working on medical countermeasures. Now, normally I'm pretty down on medical countermeasures for competently engineered threats, just because I think of living systems as being fundamentally hardware insecure. And in computer science, hardware insecure system is one that has a hardware level vulnerability, which means you can update the anti-malware all you want. It doesn't matter. It's a hardware level vulnerability. You can't change it without changing the hardware. And if the adversary compromises the system, you can't just add in new software in order to reliably fix it. The adversary can attack via the hardware vulnerability and break it such that it is irretrievably broken. If this is true for biology, then we're in trouble, because if the adversary can inject the nucleic acids from the pathogen into us, then it's plausible that there is just no vaccine that can stop it, and there is no way that we can develop some drug that will save the person in time. But that's quite a lot of assumption. If you have the whole scientific community trying to diffuse this thing, which has a lot of design constraints on it to begin with, and the adversary has to give us a good long time delay before symptoms first appear, which means if we spot it early, we have that entire time to try to figure out something that would work. And so that, I guess, is the optimistic take, that most of the life sciences community would drop everything, much like during COVID, to work on this, even if the rest of the world was just going on as normal, because it would just be that scary, and I think it would be that apparent from the genome sequence. I could be wrong, but I think it would be that apparent. And that would be our best hope, because I think it'd be hard to get the rest of the world to take it seriously. If you have good ideas about how to persuade people, I'd love to hear them.
Do we have this sort of screening in place? You know, are we routinely screening for things that look edited in the large population, or is this not happening?
So a few years back, my lab at MIT spun out a nonprofit called Secure Bio, which does run a detection system that specifically does this. So of, I think, all untargeted metagenomic sequencing, looking at wastewater and human samples and so forth, they do more than half of all of it in the world and analyze all of it looking for signs of stealth pandemics. So now we're in not perfect shape. It's not as comprehensive as it would like. Obviously, the detection is not perfect yet, but it's getting there. And given more funding, it could get to the point of being pretty close to perfect. That is, this is an area that is pretty defense dominant, the detection sphere. It's just, there's only so many things that are spreading exponentially through the human population. If you monitor all the nucleic acids spreading through the human population, if the adversary has made something, it's going to show up on that scan. They can't not have it show up if you're actually monitoring all the nucleic acids. So
can I ask you about it? What do you actually detect then? Do you detect, do you know when a new influenza is coming through? Have you ever detected something that looks like it's CRISPR edited? What are you actually finding in practice?
So I'm not currently involved in Secure Bio. They're doing a great job on their own. But yes, they have detected a number of lab created constructs that were clearly CRISPR edited. So, a lot of the time viruses are useful as gene delivery vehicles and the like, either for clinical use, but much more often just for laboratory use. You want to engineer a virus to deliver some transgene into some cell line in the laboratory, or maybe into an animal model or something like that. But if you use a gene therapy vector that's based on a human pathogen, then the secure biodetection system will spot that, and it will flag that as being an engineered human pathogen or close to human pathogen. And so anytime someone fails to adequately bleach their laboratory sample before they pour it down the drain, then the wastewater sequencing picks up that engineered construct and says, this is an engineered thing that looks like it's based on a human pathogen that was not there last month. We need to check it over. And in every case to date, it has in fact been a laboratory generated construct for a legitimate laboratory purpose.
Okay, so you can actually track where it came from. You have an idea of what it does as well. You can tell whether it's actually going to be pathogenic. And in each case, it hasn't been to date.
That's right. To date, that has always been the case. And similarly, if there's like a rearrangement in a natural virus or some new variation, those will also show up. Although the algorithms are tuned to largely ignore, single mutations or pairs or even three mutations in, say, the viral entry protein, because those mutate really fast. Otherwise you'd see every new variant of influenza or SARS-2 or the other coronaviruses.
And you're taking samples from the waters in around Boston. Is that where this is being done? Like, what are you sampling?
It's not just Boston. It's, I think at this point, I'm not sure. I don't have that most updated picture, but last time I checked, it was something like Boston, New York, Chicago, LA, San Francisco, and a bunch of others in the middle of the country. But still only US. And it would be lovely if, and then also a bunch of nasal swab samples in addition to the wastewater. And they're looking at using blood and tissue samples from medical discards to sequence and monitor those as well. It'd be lovely to get overseas personnel, particularly nasal swabs overseas, just because respiratory pathogens are the most dangerous. So nasal swabs are the most sensitive. You don't need to know who the person is. In fact, it's better not to for privacy reasons. You just want a nasal swab, totally anonymous. So it'd be really great to get those. At least I hope the US government would have overseas personnel, in whether state or defense, just periodically send in nasal swabs to be sequenced, because then that could be our forward warning. But honestly, the global air travel is such that any pandemic is going to spread to hit one of those major US cities pretty quickly. And so right now, the system's goal is to detect anything before 1 in 1,000 people is infected in any of those cities.
I see. And then how quickly can we actually move to build vaccines? And what are the bottlenecks there? Is it scientific expertise or just production at the end of the day?
It's mostly production at the end of the day. So COVID vaccines were designed within 48 hours. And now we can do it even faster. Note, we didn't really need to study the live intact viruses from that family earlier. It was mainly structural biology and vaccinology. That is, how do you stabilize the viral entry protein such that you can crystallize it and have it to be displayed to the immune system in basically the appropriate fold in the infection cycle, which, again, you don't need to study the wild type virus. You can just know how it works for the family. And so because we did know from studies of SARS-1, then just did the same thing for SARS-2. And it worked out of the box. Now, you can't always assume it's going to work out of the box. And vaccines aren't always, of course, perfectly protective. But the design is really, really quick. You can make all of your design candidates extremely swiftly, usually to the point where you can have purified test sample vaccine within a week. So if we really cared, we would then immediately go straight to human challenge trials if there was an agent that was being transmitted. So in the stealth pandemic case, if I were completely in charge and we waived all the regulations, I would immediately upon detection have somebody make a version of the stealth pathogen that is missing the modifications that we think makes it look dangerous. So take the neutered version of that, but otherwise the thing that's circulating. And then run an immediate challenge trial of human volunteers with the vaccines at scale immediately, and then scale up vaccine production. The problem is the logistics. It's just it takes time to manufacture hundreds of millions of doses of vaccine. And then it takes even more time to get them out and into people's arms. And then that's quite apart from the problem of having people support you putting things into their arms, especially if no one is in the hospital and no one has died. But that is a potentially viable solution. It's not necessarily an extremely reliable solution because, of course, the vaccine for COVID was effective at preventing illness, but it was not effective at preventing infection. And that is not reliable against a competently designed weapon. But it would help. Don't get me wrong. It would very much help.
So then when it comes to safeguards, are there meaningful equivalents of like nuclear treaties in the biosecurity domain? Do we have these sorts of things in place?
So, sorry, I am laughing out of partial despair. So there is a Biological Weapons and Toxins Convention that was signed in the 70s. And Matt Messelson, who is still at Harvard, and I've met him, and the man is a saint. And yeah, absolutely amazing. Negotiated that. And it is a pretty comprehensive ban on any development or certainly use of biological weapons. The problem is Article 10, which basically says anything for defensive or health purposes is permitted, which pretty much means if you cite some sort of for a dual use experiment, you just have to say, well, we're doing it for the beneficial reason. And then you can do whatever you want. And virtually everything in biology is dual use. So pretty much any weaponization experiment, no matter how overt, you can come up with some kind of beneficial use that requires you to run that experiment. It's just a matter of storytelling. And I'm an evolutionary biologist, so evolutionary just so stories is what we do. We're very good at storytelling. So it's not a very great treaty in that respect. You can always justify your way out of it, but you also don't have to because there is no verification mechanism. The treaty has no teeth. And to folks who always say, well, who would actually do this? Well, at the nation state level, I'm afraid that the Soviet Union had a biological weapons program that in the 1980s, which you will note is after they signed the convention, employed tens of thousands of people to mass produce known pathogens and to engineer them to be more lethal or more readily delivered by missile. And they produced smallpox in quantities of metric tons.
Does this still exist?
If you're asking whether Russia has a bioweapons program, the US and many other countries have asserted that, in fact, yes, Russia does, and North Korea both have active bioweapons programs.
And they still have these supplies of metric tons of smallpox or some sort of variant.
So we don't know on that. We know that they did testing on Vorozdeniye Island in the Aral Sea. We know that there was actually an accident where a marine biology research vessel, which was supposed to stay 25 kilometers away from the island at all times, approached to within 8 kilometers on the day of a test. And remember, they were delivering smallpox detonated via missile and then checking to see whether animals, including great apes on the island, were infected to see how well it worked. And 8 kilometers offshore, a marine biologist on deck who had been vaccinated, but the Soviet vaccines weren't very good back then, became infected. And then she actually infected a number of other people. And so 11 people were infected, 3 of them died, the 3 that had not been vaccinated. And the vaccinated ones got sick, which again isn't supposed to happen, but the Soviet vaccine was not very good. So they actually shut down all the transportation out of the region to make sure that they didn't have a new smallpox outbreak when that happened. But that was in the 70s when most people were vaccinated, and so thankfully it did not spread further. And in the 90s, Gorbachev arranged for most of the materials that they produced to be destroyed or buried. And a lot of the facilities for production were in Kazakhstan, which very sensibly didn't want anything to do with this. And so they invited the United States to come in and help them destroy it, because there was a factory that was designed to produce hundreds of metric tons of anthrax every year. And now it's been destroyed. And we know that they buried a lot of the samples on Borazdeny Island. And inspectors went in afterwards and checked the satellite images for where there had been digging and took samples and confirmed that, yep, a lot of biological material from the weapons program was buried there. So they don't have as much as they used to, but of course, this was a program in the 1980s. What was the state of bioengineering in the 1980s? Really not very sophisticated.
When it comes to producing smallpox from scratch, you said the problem at the beginning, is that it has a very long genome. But could we make it smaller? You know, could someone take the active parts of the genome and produce something that's much more easy to print out, let's say?
It's a great question. Happily, we don't understand it well enough to do that. Mm-hmm. There's definitely some genes that don't seem important for working with it in the laboratory. But for smallpox, there's only 2 labs in the world that are permitted to work with it. That's the CDC and Vector in Russia. No one else is supposed to have stocks at all. Although a number of years back, they were cleaning out a 50-year-old freezer in NIH and found smallpox samples in it. Mm-hmm. So that was a bit of a 5-alarm fire. But it is probably not the case that you can meaningfully condense a virus, because even if you know something does not seem important for study in the laboratory, that does not mean it's unimportant for, say, transmission in a population. Mm-hmm. And again, we don't have very good data on this, so we just don't have good models as to what you can and cannot delete. A broader way of saying this is, anytime a scientist tells you that this or that gene is particularly important for transmissibility, they're probably mistaken.
Mm-hmm. And then at the level of PPE and other protection, my English. At the level of PPE and other protection measures, what do we have in stock? You know, does the US have enough on hand that it can protect the entire population, or what do we have?
We have almost nothing. We have less than we did before COVID. And this is not just the US. This is pretty much every nation on Earth has declined to rebuild the stockpiles that were depleted during COVID.
Why?
Maybe you can tell me. I don't know. It doesn't make any sense from where I'm standing.
You said before the conversation that you were particularly interested in PPE, the PPE story in Singapore and Australia. What were you getting at there?
So, okay, I've spent, warning, biased bitterness flag. I spent the last 7 or so years telling everyone from every government who would listen to me that we really need much better PPE, and we really need to take engineered biothreats seriously, particularly pandemic class weapons, because when they appear, they're going to be unbelievably nasty, and you are not going to want to rely on vaccines or medical countermeasures, because against something competently designed, those are not going to be effective. And even if they were effective, vaccines are too slow. Against something that's deliberately released across airports, it will just spread too quickly before we can get that vaccine produced and the logistics solved. So what we need is to have PPE available in advance, at minimum for all critical workers. Because you can imagine, in principle, if you have enough for the critical workers, food, water, energy, communications, and law enforcement, which in a pinch the military can do, everybody else can lock down. Now, it's not strictly speaking true, because if you shut down the healthcare system, you're probably going to lose 5 to 10% of your population. And this is unfathomable to normal people. But it is possible to imagine an engineered pandemic that would make losing 5 to 10% of your population in exchange for not losing everything look like a good deal. So, but let's say, my wife is a doctor, so I'm selfish here. Let's say we actually have enough PPE for healthcare workers too, life-saving workers. So critical and life-saving workers. How many is that? Problem is we don't really know. During COVID, we designated all kinds of people essential workers, including like woodworkers who make kitchen cabinets were designated essential during COVID. That's one of my favorite examples. Probably it's a roughly 1 in 10 residents in most developed nations are the folks who would need PPE at the outset. What do we have? We do have some. We have a bunch of N95 still. 3M's N95 auras are quite good. So if you can't get something like an elastomeric respirator, or better yet, a powered air purifying respirator, which has like the lawnmower sound of pumping forced air through a HEPA filter, so you only breathe HEPA filtered air, that's the gold standard. If you have a PAPR, you are totally safe. Basically nothing gets through a HEPA filter.
Do you have these on hand? Have you got your own stockpile for your family?
I don't have powered air purifying respirators because they're battery dependent and they sound like lawnmowers. So instead, we do have elastomeric respirators. So these are ones that seal to your face. If this ever happens, I'm going to have to shave the facial hair because that is not conducive to a good seal. And they're not reliable enough for the worst possible things. But there is now an org called ProEquip, which has hired away the former executives at 3M's respirator programs, best respirator designers in the world. And they are working to build a pandemic-proof respirator. That is, and I'm always badgering them about how good it has to be, because it needs to be so good that, well, you and I are not essential workers, right? Yeah. We're thinkers. We are not actually important in a pandemic at all. But if you imagine that you were being asked to go out and work at your food processing plant or delivering food and so forth, and needed to interact with other humans, and there was something as contagious as measles and more lethal than the Black Death circulating, what level of confidence would you need in this PPE that your boss tells you you can wear and you'll be safe? What do you need to believe? It doesn't just need to keep you safe. It needs to convince you that you will in fact be safe. Because in particular, if you're not sleeping on the floor of your factory, you're probably going back home to your family. So it's not just your life on the line, it's your family's lives on the line. So you need to be really, really confident that this thing is good enough, which is why I keep advocating that it needs, at minimum, the ability to check whether your seal is working in a way that you can understand and believe works immediately, which ideally is like odorant based, such that you put the thing on, you activate it, and then to test it, you like pry up the edge just a little bit while you're breathing. And when you do that, you should smell the indicator immediately that says that there's a seal breach. So you should be able to. It doesn't look like it actually is going to be. Probably can make them for $15 to $20 each.
But when you need, in America, 30 million, you need to cover 30 million people every day for the duration of the pandemic, which last time was like 3 years.
So the plan is you really need to stockpile enough of them for the critical workers on, not exactly day one because the pandemic ramps up over time, but at the beginning, you need to have enough for all of your immediate critical workers, and you need to start making more at that point. Because first tier critical workers are those that, yes, food, water, power, law enforcement, but over time, equipment breaks down, stockpiles of existing things get used up. All of a sudden, there's a new set of critical workers who were not critical on day one, but they are critical on day 35. And so you need to have PPE for them by day 35. So you need to be able to manufacture enough for your new critical workers as more and more and more of the workforce becomes critical over time. But eventually you will have enough for the whole population and everyone can come out, albeit wearing PPE, until you eradicate it from your nation's borders. Now, note that earlier we were talking about a stealth pandemic. That's probably hard to build, right? How do you do something like that with that kind of long latency delay hard to do. I've just been saying, you asked, can you just condense the genome? And I said no, because we don't know enough about which genes are important and which ones aren't. So try to do something complicated like that. I'm not saying it's impossible. I think it's very possible. I'm just saying it's not easy. If I thought it was impossible, I wouldn't be raising the alarm over it. And if I thought it was so easy that even raising the prospect would make all of my colleagues immediately see how to do it, then I similarly would not be raising the alarm. So it's in between those, but it's probably a lot harder than just making something that is super contagious and super lethal. So I call that a wildfire pandemic. And I think it's probably the most likely thing. So the mission of ProEquip is to ensure that enough critical workers have pandemic-proof PPE to withstand the most contagious known pathogen engineered to be arbitrarily lethal or worse. Mm-hmm. And to ensure that there's enough stockpiles and that they will end up in the hands of the critical workers who will believe that they will be effective and therefore will be willing to trust their lives and their families' lives. That's the bar.
So earlier on in the conversation, you said the plague and myxomatosis are particularly deadly because they have a transmission vector that is fleas, which isn't these days that deadly to us because no one has fleas. But are there vectors that I'm not thinking about which could be used other than fleas, which could make a pandemic particularly deadly?
Not really for most humans in most middle or high income countries, for sure, and even most low income countries. These days, we just don't have parasites the way we used to. So you'd have to, suffice to say, there are reasons why I am concerned, and that is not one of them.
I'm thinking mosquitoes or something along these lines.
Oh, you could do that. So now we're going to jump ahead to gene drive is the risk. So I first
before gene drives, can I just I want to jump onto gene drives in a second because that's something that's very interesting. But can I just wrap up this topic by asking, if you were to put in place your ideal biosecurity measures, what are governments not doing that they should be doing when it just comes to regular pathogens, not gene drives?
Well, the single most important thing that government should be doing for resilience is probably to clarify that their power of eminent domain, of just seizing stuff, redirecting stuff in an emergency, that if they seize or redirect goods that were to be sent to some other party in exchange for some amount of money in a crisis that matches the crisis that is causing them to do the seizure, then they will compensate the producer for the amount of the money in the contract. Does that make sense?
So in other words, if they requisition a bunch of masks that are on way to some other country, that money flows in the direction of the manufacturer. Why is this important?
Because right now, the jurisprudence, the court decisions record in virtually every country in the world says that just compensation, in the United States, just compensation is the language in the Constitution, is defined as normal market price for the goods in question, which they have interpreted to mean normal times market price. That is the market price of the goods not in a crisis, which is not, of course, the relevant market price. The relevant market price is the price that you would have to pay for those goods in the crisis. So if you think that some sort of catastrophe is more likely than the rest of the market, you should be able to invest in a company that produces and stockpiles goods that will mitigate that catastrophe if it happens, because you are hedging against that catastrophe. You are ensuring that even as the rest of your portfolio tanks, this investment will skyrocket because you will have produced goods that other people will be willing to pay a crap load of money for.
I see. So you're saying it's not worth investing because the government can come in and requisition all of the stock and you don't make your money at the end of the day. And so that reduces the amount of investment in PPE and other sorts of defensive measures.
And across every sector of society. Why don't we have large power transformers in reserve? Well, because electric utilities hate the idea, because then they have to pay for them. Now, you would think that they could even, when they're decommissioning an old one that only has a few years of life left in it, which is why they're swapping it out, you think they could at least keep the old one as a reserve just in case something bad happened, right? But they don't even want to do that because they're big and there's storage costs. And so instead they melt them down. But if there was any kind of resilience market for large power transformers or anything else, if you could be ensured that the storage was worth it because you thought that the likelihood of, say, a Carrington event that fries a decent fraction of large power transformers, that kind of solar flare, is reasonably likely, as in fact we have archaeological data and historical data that says that it is, then you should be able to invest in a hedge against a Carrington event. And this is important because we know from COVID that governments do not prepare for low probability, high consequence events. They assure us that they will, and then they don't. This is very consistent. So instead, let the market do it. And the reason the market is not doing it right now is because everyone knows perfectly well that in a catastrophe, the government requisitions whatever it needs to fix the catastrophe. So if you anticipate the catastrophe and you make and stockpile a lot of goods to sell at elevated prices, the government will call that profiteering and seize your stuff and pay you the amount it cost you to make it in the first place. So then you are actually out your invested capital and everything you paid for storage for the intervening time. You took on all this risk and you got compensated nothing, so nobody does it, so we don't have resilience markets. So bizarrely enough, the single most important thing that governments can probably do for biosecurity and resilience in general is pass that kind of legislation. Just say, look, if you have a contract signed pre-crisis that says you will exchange goods for price X, and then we seize your goods in the crisis that matches the contract, we will pay you price X as compensation. Mm-hmm. That's it. That's the single most important thing they can probably do. Because then the private market can do all of this stuff that we're currently desperately pleading with governments to do.
I see.
I'm sounding like an American by saying, oh yes, private markets are the answer to everything.
The free market fixes all
in this
problems.
Case,
and that's actually a very small move that the government would have to make, mm-hmm. right? I mean, it's no huge investment, it's no stockpiling, it's no tech development. It's literally just change a bit of legislation.
Yep, that's right.
Let's jump into gene drives. So you're, I suppose, famous for being one of the people who built the first or designed the first practical gene drives, right? So what is a gene drive to start with?
So a gene drive is any nucleic acid string that makes it more likely that it will be inherited than normal. So a normal gene, if you have something that replicates asexually like a bacterium, gets replicated once every time the bacterium divides. If it's in a sexually reproducing organism, where you have males and females, then if it's you normally have 2 copies, one on each chromosome, and each copy has a 50% chance of being transmitted to the next generation. Well, a gene drive cheats in both of those cases. So in the bacterium, one way it can cheat is if it's like a jumping gene. So if it makes a copy of itself and inserts it somewhere else in the genome, then all of a sudden there's not 1, there's 2. And so when the bacterium divides, there's going to be now 2 copies, where before there was 1 copy. And there's various ways it can do it. It can actually just directly make a copy, or it can jump from somewhere, you can imagine after the DNA replication machinery goes past. And so now there's basically 2 copies. If one of them then jumps ahead of the replication machinery, so then it gets replicated again per that replication cycle, then even a cut and paste jumping gene gets replicated an extra time. And so this is the most common form of gene drive, and it's present in all living organisms. And in fact, in our genome, I don't have an exact number, so don't hold me to this, but it's almost half of our genome. It's some obscenely large fraction of our genome is basically broken gene drive remnants.
This is interesting, because I was going to ask you why these don't proliferate and just take over our genome. And they do, in fact.
Mm-hmm. They absolutely do. And you find more of them in comparatively larger genomes just because we don't pay as much of a fraction of metabolic cost for maintaining a large genome as something like a bacterium, let alone a virus is, right? The faster you replicate, then there's a lot of pressure to have a smaller genome, because then genome replication time is not limiting. So this is why it's actually hard to engineer a virus to add extra genes, because if you just increase the total genome size by 5%, then that means replication time is 105% of what it was before. Mm-hmm. And that really adds up over generations. That's a major fitness cost. So this is why a jumping gene does not do very well in a virus, because it's too costly to the normal virus's method of replication. So natural selection will weed them out over time. Does that make sense?
So when it comes, I asked earlier if we could condense a virus like the smallpox, and you're saying there are not so many of these self-replicating gene drives inside of a virus because it's just costly. And so I was going to ask if maybe we could just target these and remove these from a virus, but there's not many of them in a virus. Whereas in a human, we might be able to condense the genome of a human dramatically.
Yes, absolutely. And bacteria are kind of in between. They're not as extremely selected for small genome size as viruses. So there are jumping genes. Almost all bacteria do have some. But as a total fraction of genome, it's usually kept below 10% or so. I can't think of a bacterium where it's higher than 10% of the genome. Whereas in eukarya like us, especially multicellular eukarya, there's just large fractions of the genome are broken jumping genes and other forms of gene drive. So jumping gene is one form, but another form that is highly relevant is a version that is known as cut and paste. So instead of jumping itself or removing itself, instead it just cuts a similar part of the genome. So if it's present in some region of the genome, then if there's another region of the genome that looks very similar, especially on both sides, but doesn't include the gene drive, then it will cut that one. And then the cell, when there's a DNA double strand break, the cell freaks out and says, oh God, we got to fix this. And it can just jam the ends together, but that often creates mutations. So the other repair strategy it uses is it looks for a similar sequence in the genome and uses that as a repair template. So if it finds the similar sequence that already has the gene drive, then it uses that as a repair template, it copies the gene drive.
Mm-hmm. Okay, so if I understand, so for every gene we have two copies, two alleles, right? And if one of those alleles carries the gene drive, then what that does is it codes some protein, is it, or something which snips up the other or damages the other pair, the other allele, the other copy of that same gene, and then the body's repair mechanism inserts the gene drive sequence itself directly into that gap. Is that the idea?
That's exactly right. That is exactly how it works.
But how do you build one of these in practice? Could you inject someone with a gene drive, or do you have to engineer like an embryo, or how do you build these in practice?
It's a great question. So pretty much any way that you can engineer the organism to begin with, you can engineer it with a gene drive. So one way of thinking about it is, suppose you want to edit some particular gene in the genome, or you want to say add a new gene just next to an existing gene, one or the other. The way you would normally do that depends on the organism to some extent in terms of which cell do you start with to edit, and then how do you turn that into an organism. So these days, if you're making, say, a mouse that's been engineered, you do this by taking a fertilized egg and you jab it with a needle that delivers the CRISPR system, which is molecular scissors that will cut the target sequence that you want to edit. And then also you deliver DNA encoding your desired change. So you're exploiting this same cut and repair pathway where the cell says, oh no, there's damage. I need to find some similar DNA sequence. And instead of finding the other chromosome, it finds one of the many copies of engineered DNA that you just injected into the cell. So then it copies that in there. Now, instead of the original sequence, it has your engineered version.
Oh, I see.
And then
During manufacture, you have just free floating DNA that can fit into that damaged slot. So I see. So you flood a cell with these snippets.
That's exactly right. And the cell has this really amazing machinery for checking to see how similar sequences are. So it just checks upstream and downstream of the damaged region and just scans other DNA looking for similarity. And then once it finds good enough similarity, it says, all right, this is good. This is probably it. Let's do the copy. And so it does. So you do this in, of course, an embryo. Then if you grow it up, let it divide a few times in the dish, then you implant it in a pseudo-pregnant female mouse, then this will continue to develop, and the resulting baby mouse will be engineered at that site with your sequence. So this is routine. This is how we make engineered mice. This is how we make most engineered animals, is this way. The old school way was you needed to make an embryonic stem cell line, and then you would edit the embryonic stem cell line, and then you would take an embryo and you would mix it with the embryonic stem cells and hope that the embryonic stem cells became part of the embryo. And then you would hope that that embryo would develop in such a way that the new edited cells would become part of the sperm or egg producing line. And so then the next generation beyond that would have your engineered change in all of the cells. So that was the old school method.
But so the thing that I don't understand is, so when during breeding, you get one copy of a gene from the father, one from the mother, and let's say the father is the one that's infected with the gene drive, then both he will always pass on the gene drive, right, during reproduction, but only one of the offspring's alleles will be infected, right? So at what point does that allele get infected? Is it at what point when I've no idea how the transmission actually happens. When does the cutting happen during breeding?
Yeah, great question. So remember, there's two different repair pathways. One is jam the ends together. Broadly speaking, there's more than two, but broadly speaking, there's jam the ends together, and then there's find a similar sequence and copy it over. So you need to have the gene drive turn on when it's maximally likely to copy over. So we call this homology-directed repair because the similar sequences on both sides, we're saying we're looking for homology, for similarity on both sides. So if you go through homology-directed repair, you copy the gene drive. If you jam the ends together, you don't. So the time when it is most likely to do homology-directed repair is the time in development when you normally shuffle the chromosomes. And that is during meiosis, when you're making sperm and eggs. So an ideal gene drive system wants to turn on just before the chromosomes are normally damaged so that they can be shuffled. Because then you're maximally likely to have homology-directed repair happen. If it happens 100% of the time and you cut 100% of the time, then any organism that started with one gene drive copy, it's going to reliably cut the other version, and it's reliably going to be copied over. And so it will then have two copies. And so every offspring will inherit one. So that's perfect 100%. Obviously, 100% never happens in the real world, but very conveniently for us, in Anopheles mosquitoes, the malaria spreading ones, it's like 99.8% is the best anyone has gotten to date, which is crazy high. Conversely, in mammals, I don't believe anyone has gotten even 35%.
I see. So then this isn't a very good weapon for against humans, right? If you wanted to engineer a gene drive as a pathogen to wipe out the human species, you're not gonna hit above 30% of the population.
No, but even if it was 100%, good luck waiting those 10,000 years and hoping that humans never, undo it with even better technology.
I see. Because we breed every 20 or 30 years, you have time to... I suppose if you infected enough people, though, initially.
Yeah, but how are you going to do that? I mean, it's like saying, how are you going to engineer the germline of a whole lot of people all at once? Seems hard. You need a horizontally transmitted agent that then infects the germline somehow. But this is what nature has evolved to prevent natural viruses from doing, because any virus that can reliably get into your germline, like that's the really reliable way to replicate, because then this is how you get broken remnants of gene drives in your genome. So this is why we have a separate germline from the rest of our body, and why the body puts a lot of barriers around the sperm and egg producing cells to try to make it really, really hard for any virus that infects the rest of us to get into our germline. So there's just a lot of barriers to making that difficult. So it's hard to imagine how you do that. But let's take the general question, because it's really important. Is gene drive an effective weapon or not? Could it be an effective weapon? Is there some way to make it an effective weapon or not? Another way to ask this question, and from military theory, is does it favor offense or defense? But the way I like thinking about it when we're talking about technologies is, does it favor disruption or does it favor stability? Now, I'm not saying stability is always good. Obviously, if you have a tyranny, then perfect stability means you can never have a revolution and you're stuck with tyranny forever, and this is bad. But in general, when you're talking about potentially extremely powerful technologies, I'm worried about those that are disruption dominant. Those that would, in military, would favor the attacker to the point where there's just no good defense. But a really disruption dominant technology is not just one that favors the attacker. It's one that's easily accessible to just about everyone. That is, proliferation is also a factor in the disruption score. So gene drive. How do you tell whether a given technology favors disruption or stability? I really wanted to know the answer to this question back in 2013. Because when I first realized, hey, we could just encode CRISPR, the same CRISPR system that we use to make the initial cut and to deliver our engineered DNA, we can just encode it with the engineered DNA so it gets copied in, programmed to turn on during meiosis just before you make sperm and eggs, and then we'd have a gene drive that would spread through the population, and we could use this to get rid of malaria and schistosomiasis and solve all these other problems by editing wild species. This is going to be amazing. And you will note that I'm assuming that, yes, it is good for us to engineer wild species. A lot of people disagree with me. Personally, I am in favor of rewriting the tapestry of life because I have some moral objections to the way that nature did it. I am deeply grateful to nature for existing. I am deeply grateful for all of the beauty that surrounds us. I respect it. I want to learn to create similar amounts of beauty, ideally without all the suffering and horror. Thus, rewrite the tapestry of life. But I also recognize that I am not the only person who lives on this world, and other people may have different opinions, and in fact do have very different opinions and intuitions about when we should and should not engineer things, which is why I think it's incredibly important that if you're even contemplating developing a technology to change the shared environment, you have to do so openly and transparently and invite people's voices on what you should and should not do while it's early enough to change the technological approach that you're taking.
Can I stop you here and ask if you, for instance, took it upon yourself to, I don't know, release some sort of a genetically modified mosquito or whatever it is that you want to release, are there laws that you could get in trouble, by breaking? What's the status there? Are there any laws in place today or not?
So they have added some research regulations on arthropod safety levels intended to keep gene drives like that from getting out. But the actual teeth on them, like how much trouble you would get in, criminal offense, etc., it's really not clear that anything particularly bad would happen to you pretty much anywhere. Now, a lot of European nations have, frankly, totally unbelievably irrational laws about all genetically engineered organisms. And in fact, when I was talking to some European regulators once, I was explaining how gene drive was different from a normal engineered organism. They said, oh, well, so what you're describing is a gene drive. We just assume that all engineered genes will do that. And I was like, you what? Why would you do that? Because normally when you edit an organism, you're changing it. You're changing its function. You're redirecting its resources to do something that it would not do in nature. Nature is the one that optimizes for replication, which means you're going to be impairing its ability to replicate, which means by default, I don't care what you're doing to it, natural selection is eventually going to weed it out. Unless you've figured out some clever way to create a fitness advantage that nature has not previously figured out, which is kind of my academic specialty. But its point is it's hard. It is not the default. It is absolutely not the default. It is extremely difficult to do. Yet a lot of European law just assumes that any given engineered organism is in fact going to be fitness positive and change the whole wild population. So in one sense, they had a leg up in terms of regulating gene drive. But even there, I'm not sure it's a criminal offense.
So let me ask then, initially you jumped onto gene drives when I asked if there were vectors available for producing pathogens that spread even, well, for fleas it's when the victim is already dead. But in this case, could we use gene drives to engineer mosquitoes which transmit highly deadly pathogens to humans? Is this where you were going initially?
So yes, so the second day after I woke up and thought, good God, if you can engineer a mosquito to never spread a pathogen, you can equally well engineer it to always spread a pathogen, or worse, with every bite. Who could do this? Is this technology dangerous or not? And so I actually didn't tell anyone until I thought about this intensively myself, did a bunch of searches trying to find a framework that would tell you whether your technology favors disruption or stability, although I didn't even have those terms for it then. And I have since actually written such a framework, but I'm actually reluctant to share it because could actually be kind of dangerous because it tells you which technologies are most disruption dominant and how to evaluate that across different scenarios. But with gene drive, I kind of tried to reason it out from first principles. So you said, can you, in fact, engineer a mosquito so it always spreads a deadly pathogen with every bite? The answer is yes. Of course you can do that. But does that make it disruption dominant? Does that make it an effective weapon? And I concluded the answer is no, for three important reasons. Number one, this is a technology that spreads slowly. It's only parents to offspring. And when you think about it, it only has an advantage when the organism has one copy of the gene drive. If an organism already has two copies, it doesn't do it any good. Obviously, if there aren't any copies, it doesn't do it any good. So if you can imagine you're dropping a gene drive into a population, it spreads outwards through the gene pool, but it only has an advantage around the edges. Mm-hmm. All the ones in the middle already have two copies, and so they're not like counting in the population where it has an advantage. So broadly speaking, it tends to increase by a factor of about 1.5 every generation. Now, mosquito generation is like 10, 12 days. Pretty fast, but still 1.5x amplification is pretty slow on the scale of things. It's not, to give you an example, like we think of gene drive in terms of how fast it can spread through, say, a mid-sized African nation because we want to use it against malaria. Okay, well, unless you release it in the middle of the African nation, it's actually going to take like a decade to spread all the way across. It's just not very fast. And you can raise mosquitoes in captivity a heck of a lot faster than that. Because if you have one gene drive mosquito in captivity, well, in captivity, you can ensure that all 200 of its offspring will survive. Whereas in the wild, on average, it's going to have two. So you can amplify that gene drive much, 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 much faster in captive mosquitoes, creating huge numbers that then you can release in the wild.
Disperse.
So this is slow relative to how fast you can do things in captivity. And it gives you plenty of time to respond. You have years if there's an offensive gene drive that's been deployed somewhere. It is obvious. So first of all, if all of a sudden every mosquito bite is lethal, I think we're going to notice pretty quick. But even if you do like a time-delayed lethal kind of thing, if you're bothering to do this kind of untargeted metagenomic sequencing, which admittedly we were not doing back in 2013, but I figured we were eventually going to have to do for the same logic that I related earlier, you will spot it. And in fact, you might say, well, when are we going around sequencing the genomes of mosquitoes? And actually we do this all the time, especially for the disease-causing ones, especially for the ones that bite humans. So we would in fact notice from the genome sequencing if there was a CRISPR system in there. Because CRISPR systems do not occur naturally in sexually reproducing organisms. They're from bacteria. So it will stick out like a sore thumb. We cannot help but see a gene drive spreading through something like a mosquito. And if you want to extend this to other organisms, well, just deep sequence the river, and you will detect everything that lives in the river and everything in the watershed that washes down to that river. That is, you can pick up mosquito DNA by sequencing a river with reasonably high sensitivity. So you sequence deeply enough, you will see everything. You would see gene drive in, no, it doesn't matter what species it is, you will spot it. So it's obvious if you look. You cannot possibly hide it. There is no way you can engineer this gene drive that it would look like anything other than a very obvious gene drive. It is slow, it is obvious, and it counters itself, which is possibly most important. So like the problem with an engineered pandemic is that the vaccine, you can't counter a pandemic with itself. A good guy with a pandemic does not actually help you against a bad guy with a pandemic. Learning how to make pandemics does not help us defend against pandemics at all. Making vaccines faster also is not very helpful because the pandemic spreads faster than the vaccine. But gene drive counters itself. It spreads just as quickly. And of course, it spreads faster in captivity. And when I say it counters itself, what do I mean? Well, CRISPR, you can program to target basically any sequence, right? So suppose someone were to engineer a mosquito that spreads a lethal illness with every bite. There's some sequence in its genome that has CRISPR for the gene drive part and then has the lethal with every bite sequence. So I'm going to take that sequence, I'm going to exactly duplicate the CRISPR part, and then I'm going to add extra instructions telling CRISPR to cut and replace the weapon part. So then my version, whenever it mates with the weapon version, the offspring is going to inherit one of each. And my version, which we call an immunizing reversal drive, is going to cut the weapon and replace it with itself.
I see. So if someone were to breed millions of mosquitoes with this weapon in the genome, then you could, as a defense, breed a million of your own with the inoculation and release those.
That's right. And in fact, we can breed a lot more than a million because if you're the defender, you usually have more resources to overproduce things. Mm-hmm. And again, their million goes from a million to 1.5 million to 2.25 million to, and so on. And as soon as I have my first one, I go from 1 to 200 to 4,000 to 800,000 and so on. So I catch up much, much, much more quickly. And
although they have the element of surprise, right?
Just as well.
They have the element of surprise. You have to work out what's going on. So if you had a malicious actor who dropped millions of mosquitoes across the United States or somewhere in Africa or somewhere anywhere in the world, really, then they could take out a large pockets of populations, right, before you detect anything.
You could, but our first weapon, of course, is insecticide or screens. Like, I basically never get bitten by mosquitoes unless it's in the summer when I'm taking a risk and I'm outdoors. Now, some parts of the world, people are more vulnerable, yes. But broadly speaking, in the United States, if you want to take precautions against getting bitten by mosquitoes, then you do. I mean, whenever Eastern equine encephalitis gets bad here in Massachusetts, like my family just doesn't go outdoors in high-risk areas. And we lather up in picaridin and DEET. And it's generally fine. We almost never get bitten. And when we're not deciding not to take chances. But more to the point, if you really want to get rid of the mosquitoes, break out the DDT. Like, we would relax the environmental laws if we were attacked by an engineered mosquito bioweapon. So I'm just not very concerned by this. Not because you couldn't use it as a weapon. Of course you could use it as a weapon. But there's a lot more effective ways to kill people than to do this. Because remember, engineering a mosquito in the first place requires you to get mosquitoes to lay a bunch of eggs, and then you have to inject them using micro-injector equipment, extremely high precision, extremely difficult. You have a very low success rate for making the transgenic mosquitoes, which typically you have to, make them glow so that you can spot them earlier. And it's most effective if you have a $500,000 flow sorter machine to detect the fluorescence and so on. And there's probably only a dozen or so labs in the world that can do it at all. Mm-hmm. So that is to say, this is probably about as accessible as making smallpox.
So at the beginning of the discussion, I asked, why haven't we seen an attack? A catastrophic attack, which has been directed and killed tens of thousands, hundreds of thousands of people. And what seems to be coming across is it's just very difficult. There's low access. People aren't cartoon villains in general. State actors don't really have any motivation to do it necessarily. It's not that we've been lucky up until now. It's just not as accessible as I had imagined. I'd imagined cartoon villains in some sense.
So that seems right to me, but keep in mind that probably 50,000 people, not 10,000, can perform reverse genetics with an influenza virus. And you might say, well, what are the most effective defenses? So another spinoff from my lab was this DNA synthesis screening system that uses cryptography to preserve the privacy of the order, and a bunch of other stuff offered for free from a Swiss nonprofit. And if everybody used it, we would not have these problems because it can detect not everything, but like I struggle to break it. Mm-hmm. And I'm pretty good at this. But most people don't use it because there's no laws requiring them to use it, except in France due to odd historical chance, or maybe the French are just farsighted that way. But long story short, we ran a test of DNA synthesis screening where we placed orders basically to check are companies doing what the US guidance said. Now, there's no law forcing them to do this at all. But we also wanted to test just for...
So can I stop you? So this is companies that generate DNA. So I can call in or I can send in a request for a sequence of DNA, and you're checking whether these companies check whether it's something pathogenic. Is this the idea?
Mm-hmm. That's right. So will they just send you DNA sufficient to create something dangerous? Mm-hmm. So offhand, what would you say is something suitably dangerous? Now, smallpox actually isn't all that dangerous because 186,000 bases is a lot. We wanted something where if this succeeded, we could get the whole thing. What's another dangerous sounding thing to you?
I mean, initially AIDS, right? But this is not what I would design if I wanted to create a pandemic.
Right. So if you think further back in history, what was the pandemic
plague,
that killed
the Spanish flu?
Bingo. So that's the one that we tried. So 1918 influenza is a select agent, which is to say it is illegal to have a sample of the virus without going through the ridiculous registration required of the select agent program. This is as it should be. The law is even good enough to recognize that the nucleic acid sufficient to generate the infectious virus is also considered a select agent. Mm-hmm. However, if you split that nucleic acid into two pieces that can be trivially assembled by any trained high school student, it is perfectly legal. So we ordered pieces of the genome of 1918 influenza, which is only 15,000 bases. So if you order it from a provider, it will probably cost you somewhere around $3,000. But we ordered enough to make it several times over, and we ordered it in pieces, and we did various other tricks to it in various ways just to test them. Are they doing what the government guidance said or not? And would they send it to us when we were using a fake name, and we actually sent it to the office address of our biosecurity nonprofit that didn't have any laboratory space? So it should be an obvious no-no. You shouldn't do that. Fake name, etc. Well, almost all of them shipped it. None of them asked questions. So things have improved since then, but it is still not illegal anywhere other than France. And this giant loophole in the select agent laws where it's illegal to have the intact thing, but if it's in two pieces, it's totally fine. And, the companies didn't do anything illegal. It's a guidance. It's a recommendation of the US government that companies do this. We have not bothered to require it.
Can I stop you for a second? At the very beginning, when I asked what do we have, what's available, you said that smallpox was the only agent that we have that you saw as being a potential for a pandemic. But then if we have the Spanish flu, is that no longer deadly, or why did you not include that?
Great question. It is not not particularly likely to cause a pandemic today. And the reason is that, so first of all, it's more than a century old. They call it the mother of all pandemics because the H1N1 influenza, which is the same sort of surface types, is still circulating today. But what's more, the 2009, they call it a pandemic, but I don't really refer to anything as a pandemic unless it kills at least 1 million people. But anyway, it was a new variant of flu that came from pigs that recombined with the human circulating version. And it has this H and N, which is the ones that the vaccines recognize, that our immune systems recognize. And that one just so happened to be really similar to the original 1918 in terms of what it looks like to the immune system. And so were the subsequent years. So a lot of the population has some cross-reactive immunity to 1918 influenza because we were exposed to that, or we got vaccinated with the seasonal flu vaccine anytime since then that uses the very similar ones. Which is to say, normally influenza is not that transmissible. And so if there's a good deal of population immunity out there already, it plausibly is not transmissible enough to spread at all. Now,
I see.
How likely is it? I wouldn't say it's like less than 5% chance.
Mm-hmm.
It might be 10% chance, like actually saying this legitimately, it might be 10%. My rule is I'm never going to say if I think it's over 10%, but it might be as high as 10%, maybe. But again, this is why we were willing to do it. This virus is dangerous enough that in expectation, you should not be able to get it. You should not be able to order it in pieces that can be trivially assembled by a high school student. You should not be able to do it without anybody noticing, without law enforcement knowing, without because if it were to cause a pandemic, then even if we, take into account all of the better drugs that we have now and antivirals and the like, even if you assume that it would not be as bad because we have population immunity, even if it wasn't good enough to stop it from spreading and infecting a lot of people, it still made things less severe. It would still be as bad as COVID. Mm-hmm. So a 10% chance of another COVID is in expectation around 2.7 million dead people and a whole lot of economic damage. So that is to say, this virus is basically the equivalent of a nuclear device.
I see.
But do we treat it as dangerous as a nuclear device? No, of course not. That would be a silly idea. But how many people actually could, given highly enriched fissile materials, build a nuclear device? 50,000? Because that's the number of people who could make infectious 1918 influenza from the DNA that we were able to obtain, totally legally.
I see, but I suppose it's less scary because the damage is dispersed, and it's mainly older people and people with compromised immune systems. I see, but so one of the other aspects of this story is then that when we don't really know how to engineer a pathogen from scratch, and so really what we have is pre-existing pathogens and edits of those, and of the pre-existing pathogens, mainly we have herd immunity already, or there's some level of immunity, and that's what makes them less dangerous. So I imagine that we had labs with stores of highly dangerous and known pathogens, but it's just that the selection is very thin, and that's the problem if you're a cartoon villain.
If you're a cartoon villain, you just don't have much ammunition right now. But the reason I'm concerned is, again, I said publicly credible. If you really know what you're doing, you have more options than that. And there are very few people who really know what they're doing in this space. My concern, and actually I used to be more concerned because I thought it was more obvious than it apparently actually is for a bunch of dangerous things that it turns out normal people don't think about because normal people apparently have filters on that I don't have. And this is why I worry about these things, and most people don't. But the problem is, as AI gets better and better, it is getting better at synthesizing ideas, especially across domains. So to give a specific example, I spent a lot of time helping to red team these systems, because mostly they focus on ensuring that the AI is not going to tell you how to design the reverse genetics constructs for 1918 influenza, how to, actually get reverse genetics to work, how to troubleshoot it, which protocol to use, which cell line, what kind of equipment and materials use, all the operational details. What worries me even more is, what if the model tells you which sequence of virus you should use that is not something obvious like 1918 influenza? What if it were to tell you some way to make a pandemic that is not obvious, that is not publicly credible, but would in fact work? What if it's something that requires you to venture, say, outside of virology? Because most scientists are narrow hedgehogs who focus on their one area. Most threats are cross-domain. So, for example, one of the models suggested a creative way to make a toxin that is probably about as potent as botulinum toxin, which is the most lethal toxin in nature, by leveraging an aspect of physiology that I did not know existed. That is to say, an AI taught me something about building bioweapons that I definitely did not know before.
And I suppose since these are unknown unknowns, then there's no defense in place at the level of controlling the LLM, right? No one's programming in that the LLM shouldn't tell you about this specific pathway.
This is my current crusade, that the models right now the guardrails are too focused on overt defenses. So they are more likely to say no to this. Like Claude is really uncomfortable talking about engineered virology in general. It will do it to some extent, but it doesn't like to. It's always on high alert. In fact, anything in what they call the CBRN class, Chem, Bio, Radiological, Nuclear, they're a little bit more on edge, a little bit more likely to trigger and decline. For the most part, you're right. They tend to be defended with separate classifiers that are just basically a dumb model that is looking for particular signatures of a particular topic that the safety team was aware of. And because this is an unknown unknown that as far as I know, no human had ever considered before that you could do this, there is no way that the safety team could possibly have known to restrict it. Mm-hmm. So what you need is the model to recognize the consequences of the innovation itself. Mm-hmm. And you might need it to prompt itself to try to run that check always. Or you might need a separate model that gets triggered when it's in a possibly dangerous territory to start reviewing it. Or, there's a bunch of ways that you could go about doing it. But ultimately what you need is the model to consider the consequences for itself and whether or not it can continue, and to greatly expand the number of trusted user programs. That is to say, if you're a scientist working in, say, virology, say you study influenza, you probably know an awful lot about influenza already. You've probably thought about all the different nasty things that influenza can do naturally, at least. And you should be able to get DNA for most, influenza strains by default. Anything that you have permission for, you should be able to get. Any question you ask an LLM about influenza, it should be willing to answer because you're an influenza biologist and that's what you're researching. You have a legitimate need to access relevant information, relevant papers, relevant protocols, get advice from the AI to do your research better. But it should not tell you anything about toxins because you are not a toxins researcher. It should not tell you about bacterial pathogens because you are not a bacterial pathogens researcher. It should tell you about stuff that is dangerous if you are a legitimate researcher in that narrow subspecialty. And what it should not tell people about is cross-domain stuff. People like me, the foxes, we are the problem because you're much more likely to come across an unknown unknown as a fox running about sniffing interesting things to see what can be combined than if you're in one well-understood field digging. Mm-hmm. Digging can come up with new stuff, but usually it's the cross-domain synthesis. And so that's where we need the AIs to be careful about assessing large-scale consequences, especially across domains, and avoid rejections to legitimate research by saying, you can ask the model anything about your area of research based on your publication record or your mentor's publication record. And even if it's a dangerous area, it should tell you because you're legit. But that does mean you need hardware key plus biometric authentication that you actually are who you say you are. Use the PKI cert system that, we developed one for DNA synthesis screening, and so that legit researchers could automatically have, an influenza lab with permission to work with influenza could automatically get any of the strains that they work with just by sending in their PKI cert with their order. Like, all the tech for this exists. That's just where we need to go with AI, but it's hard because you need to convince the company that they have a big enough problem to do this. And it's also a little bit unfair because we don't actually have identity verification. My understanding is Europe is going to come up with a really good online identity verification system rolling out in like a year or so. Like Denmark already has one, Estonia has one. So European scientists could presumably use that to log in to recognize their ORCID account and have all their publication records scanned, and then they would get a PKI cert whenever they're logged into their verified system, and then they could use that as their base ID for the PKI cert rather than the centralized server that then they send to the model they're talking with, the DNA synthesis firm, whatever. And then that way legitimate research could proceed unimpeded, and we would actually have some barriers against models disclosing very nasty things. Because right now the model doesn't really know what might and might not let you make a wildfire pandemic. Or rather, the model might actually know how to do it, but the model is not going to recognize on the fly that what it is just disclosing is in fact the key to doing it right now. We can change that.
So from what I gather, the main threat that you see today, at least in the short term, comes from AI and its interaction with biosecurity. What do you wish people understood about biosecurity that they're not really understanding? Like, what is the one message that you would like people to understand when it comes to this interaction that we're not really seeing yet?
That biosecurity is a problem of insecure hardware and network security. That is, assume that a competent adversary that can inject nucleic acids into you, that's it. Don't rely on vaccines because that's anti-malware protection. Don't rely on medical countermeasures because that's trying to scrub the system of malefactor influence after the fact. Those don't work in cybersecurity. They don't work here. But conversely, for all human-to-human stuff, we didn't really get into mirror life, but for all human-to-human stuff, it relies on nucleic acid transmission from human to human. We don't need to allow that. So the long-term solution, the immediate solution is have enough stockpiled pandemic-proof PPE such that eventually everyone can wear it. And then we are immune to obvious pandemics like wildfire, but not stealth because you may not be able to convince everyone to wear it. Hopefully we can, but it's not guaranteed. But the real long-term defense, shut down the network. End all infectious disease, which we can do if we just engineer the built environment. Indoors is where almost all respiratory stuff spreads, not outdoors. Remember, if you're outdoors in COVID, you're fine. And COVID ended up being quite contagious. But still, outdoors, you're largely fine. Don't speak in really close proximity to someone outdoors, but broadly speaking, you're fine. And indoors, that just means install far UVC lights, which are germicidal. Install vaporizers for triethylene glycol, which is a generally recognized safe substance in a lot of our foods and products and so on. It's also quite germicidal in the air. And even the new influencer thing is hypochlorous acid, which is, I don't know, in a lot of products apparently as well, but it also looks like it might be germicidal in the air. And you can probably put that in a normal cool mist humidifier, ultrasonic kind, and just mist in the rooms, and it'll kill airborne pathogens. So if we were to do all of these things, we can just shut down unauthorized nucleic acid transmission permanently. And then we would not have to deal with infectious disease, and your little one would get you sick a lot less in the future. But we would also be immune to all pandemics, natural and engineered. So that's the long-term solution, network security.
So if I understand correctly, we actually have the technology in place to end all contagious disease, and the technology isn't at the level of vaccines, but rather just engineer the built environment.
That's right. The answer to biosecurity is not found within biotechnology because living systems are hardware insecure. We'd have to rewrite ourselves at the hardware level, just completely changing the biology of how our cells talk to each other and sense and respond to threats. And that's just not a feasible thing. But if you just change the built environment such that it's not viable to have a nucleic acid be transmitted from person to person, then we're immune to pandemics and basically all infectious disease. Now, would this be costly? Yeah, it would be costly. Right? The cost is coming down. There's a lot of people developing new solid state emitters for far UVC light. So 222 nanometers. It's really great because it gets absorbed by proteins. So hits the surface of our skin, just gets absorbed. The outer layer of cells in our skin and eyes is usually dead or sloughs off within 24 hours anyway. So they just basically take the beating, all the proteins get absorbed, DNA doesn't get replicated anyway. So basically, if you're multicellular, you're fine. But if you're a virus or a bacterium, you need the proteins in that virion or cell. You can't afford for them to absorb all of this UV and get broken. You get inactivated. So it's a preferentially multicellular selector. If you're multicellular, you're fine. If you're not, you're toast.
And so it's just UV radiation inside public spaces.
Yeah. And it's appallingly safe. Like the only complication is it makes a little bit of ozone. But the amount of ozone that it makes to have it work in a room, even before you add like ozone descrubbers, ozone decomposition catalyst tech, you typically get more ozone when you open a window. The levels outside are usually
but
higher.
You could roll this out in aircraft, for instance, or I mean, you don't have to roll it out everywhere. You could probably cut down huge. You could cut down the amount of sickness in the population by being extraordinarily selective of where you roll these sort of this technology out.
Yeah, absolutely. And you could save a ton of money because when you, at least back of the envelope, the amount of lost productivity just due to, yes, people calling in sick, but also people just being under the weather and therefore less productive. There's been a lot of literature on that just shows, yeah, when you're sick, like even if you try to work, go into work, you're just not as effective as when you're not sick. And then you add the long-term chronic disease burden from conditions that are triggered by an infectious disease, which is a lot of them. A lot of chronic diseases is because some pathogen came in and screwed something up in your body, or your immune system became overactive in response to the pathogen and screwed something up in your body. And then you live with the effects of that for the rest of your life. That's what long COVID is. It's not one thing. There's a bunch of different flavors of long COVID depending on which system got screwed up. And COVID isn't unusual in that way. This is just what pathogens do. It's very rare in any one person, but it adds up because we get infected with so many different things in our lives. So it would be super cost effective. It would certainly pay for itself if we could roll it out inexpensively enough. And triethylene glycol in particular is just really, really, really cheap. And far UV, some people are wary of it just because it would probably discolor some of our fabrics. If you think about the UV bleaching effect. Yeah. But
so it's not a skin cancer risk.
Mm-mm. Nope, not a skin cancer risk, not an eye cancer risk, not an eye damage risk. Like broadly speaking, we wrote a review a couple years ago, just looking at all of the known safety effects and what are the gaps for far UVC. And it's just really safe. The levels that have been approved for use by the ACGIH, which is the North American Council on Industrial Hygiene, I think it is. But anyway, they do like set workplace safety levels that then, the national authorities like OSHA in the United States normally just uses whatever value they recommend. And the maximum level that they permit is enough to drop the number of infectious particles in the air by 99% if we were to use that. So it's already legal. It's already regulatorily permitted. We just need cheap enough solid state emitters that then you can just imagine you install them in the light bulbs everywhere, along with the ozone scrubber. And that's it. We're golden once we can get to that point. So this is why I'm ultimately an optimist. Like, yes, I think you can absolutely engineer a wildfire pandemic, R naught 12 plus, highly, highly, highly lethal. I think you can absolutely harder, but engineer a stealth pandemic. that has a year-long delay or what have you. And I think we will absolutely learn how to engineer the surface of any given existing virus to completely evade population immunity. Because when you think about the number of possible combinations of sequences that have that same function of the existing one, but are different enough that our immune system is not going to recognize it, sequence space is vast. Just the number of combinatorial possibilities is so large that even a tiny, tiny, tiny fraction that have the same function is still a lot of various sequences. So to bring it back to Feynman, the contrapositive, what I understand, I can create. We will understand how to design proteins well enough to duplicate the function of any given surface or entry protein on a pathogen, enough that we're going to be able to reskin that pathogen and make it as infectious in terms of its effective R as its R naught, its theoretical R in a naive population. We're going to be able to make pathogens that treat us all as naive without changing the genes, right? Other than those entry proteins. To your point about adding or deleting genes, you wouldn't do that. You would just change the surface ones by enough that it would evade our existing immunity. And then if you add that separately with insights into how do you make it do nasty things, that's where the threats actually live, is the combination of those capabilities. And I just cannot imagine a future where we don't acquire that knowledge. Because again, I want to rewrite the tapestry of life. We're going to have to learn things like that. We can delay it, but eventually we're going to go there. We can't delay it forever. We will learn how to create catastrophic pandemics. We are going to be dumb enough to disclose how to do it. Once we are dumb enough to disclose how to do it, the scientific community is going to be dumb enough to argue about whether or not it would work. And then we will make it credible. And we will have made all the recipes and blueprints public. And the question is, at that point, will we have actually done something about DNA synthesis screening? Will we have enough pandemic-proof PPE for critical workers, and where will it be? And to bring back that point about Australia, Singapore, I think we should roll them out first in Singapore as the pilot, and then Australia, and only then the United States. And previously, I just assumed United States because I'm American, that's where we are. The US is an energy exporter and food exporter, so obviously we could sustain ourselves if we can survive. But I was neglecting, thinking about this for a decade. Only recently did I consider, wait a minute. I can't just think about this problem just from the biology perspective. What are our adversaries going to do if there is a clearly engineered wildfire pandemic spreading around the world and the United States is surviving because it stockpiled pandemic-proof PPE in advance? Now, I don't care that it's not the US government stockpiling the PPE. They're going to look at this and think, how many people associated with this project, have advised the US government on pandemics, have had clearance at some point in their lives, but more to the point, smoking gun is they were protected and we weren't. And then the weapon appeared and our nation is disintegrating. They have won. They will inherit the earth. We lose. Are they really not going to push the button? I would like to think of human nature that they would not push the button out of spite and vengeance. But they can't know that the US didn't make it. They can't. They're always going to be suspicious that the US made it or arranged for it to be made or was just aware of some terrorist group that was going to make it and let them do it. So we can't rely on them not hitting the button, which means, unfortunately, you have to assume counter-value exchange. Basically, the whole Northern Hemisphere is gone. Therefore, the respirators need to go to Singapore and Australia first, because neither of them can survive on their own. But together, their supply chains are extremely complementary. So in the limit, as technology gets more destructive, this probably will not happen, of course. Probably no one would make a wildfire pandemic in any given period of time when it's going to be relevant. But the chance is definitely not zero. And it would be very foolish of us to stay unprepared, given the cost of losing everything if we fail. So I think we must take precautions to protect the light of civilization from the worst possible threats, which someday we will know how to make.
I didn't realize Australia was part of that key. On that note, it's been a pleasure, Kevin. Thank you for coming on the podcast.
Thank you, Shane.